Okay, guys, let's have a quick look at these here. So for number 47 there, we got manganese 2 oxide, ionic compound. We see a Roman numeral, it's automatically an ionic compound. Okay, so we write the formula for the metal, which in this case is manganese. This is the manganese that is 2 plus. It is with oxygen that is always 2 minus. So that compound is already balanced and should just be MnO. Okay, if you wrote it as Mn2O2, remember you always have to reduce down to the lowest terms. Okay, for number 49, we got mercury 2 nitride. Again, we see a Roman numeral, so we know it's ionic. We write the name or the symbol for mercury, and this one tells us it's the kind that's 2 plus, and it is with nitride, which is always a -3. So we're looking at Hg3N2 for that one after the drop and swap. Okay, if you got questions on these as we go, guys, please ask. Okay. Then we've got lead 2 hydroxide. All right, so lead is Pb. Okay, this is the 2 plus kind of lead, and it is with hydroxide, which is a polyatomic ion that has a -1 charge. So when I drop and swap here, okay, the 2 will end up here, and I'll need what? Brackets. I don't know why that one bracket is so much bigger than the other, but that's just the way it happened. Okay. All right. And then for number 52, we got selenium tetrafluoride. As soon as I see a prefix, I know this is what kind of compound? Molecular. All right, so I'm going to write selenium, Se. It doesn't have a number, so hey, there must not be more than one of them. And then tetrafluoride means there's how many fluorines? 4. SeF4. And then we got phosphorus pentabromide. Again, we see a prefix there. So I write phosphorus, there's no number beside it, okay, and it is pentabromide. So how many bromines are we looking at? 5. All right, and then we have nickel 2 sulfate. We got a metal, it's got a Roman numeral, it's definitely ionic. Okay, so we got nickel, it's the 2 plus kind because that's what the Roman numeral says. And it is with sulfate, SO4, it's a -2. Those already add up to 0. So that compound is fine either this way or enclosed in brackets. Either way. Okay. And number 56, we got silicon dioxide. Okay, it's molecular because I see a prefix there. There's no prefix on silicon, so it must be a 1. And dioxide means how many oxygens? 2. Okay, and then we have lithium phosphate. Okay, is that ionic or molecular? Ionic. It's ionic. Lithium is a metal. Okay, so Li plus 1. Okay, and phosphate, PO4, polyatomic ion with a -3 charge. When I drop and swap, I'm going to have Li3PO4. PO4 can be enclosed in brackets, but it's not necessary. Okay, and the last one you guys had to do on here was cadmium nitrate. So cadmium is Cd, right? It's not one that comes up very often. Okay, and what's its charge? I don't even remember where it's at. Oh, there it is, 2 plus. Okay, Cd. Here's a generation gap question for you. What does that stand for, CD? Compact disc. Nicely done. Hey, most of you are going, I don't even know. Never heard of that thing. Those were so cool because you didn't have to have a tape deck anymore. You guys probably don't even know what that is. Okay. Used to carry around this thing called a Walkman, and it had a tape deck, and it took like 4 AA batteries, lasted about an hour and a half, and you had to put 4 more in. Okay. Yeah. You have it so good. I walked to school in the snow up to my hips, both ways uphill. Yeah, okay. So, okay, cadmium nitrate, NO2. Okay, so we got cadmium with the 2 plus charge, nitrite with the -1 charge, so we definitely need brackets here, a 2 outside of brackets. Okay. All right, questions on any of those? Okay. I'm going to put up a couple to go the other way, formula to name, not as many, okay? Practice those for a little bit, and then we're going to talk about lab reports. I've already put something in Google Classroom, okay, new this morning. It's just a template, Okay, kind of shows you what the expectations are. We're going to walk through that, okay, so that you have an idea of what you'll be in for when you're writing lab reports for me. Oh, come on. All right, so I would like you guys to name for me number 3, number 4, number 5, number 8, that's an easy one. Number 11 and number 12. Okay, you got 6 there. Give you a couple minutes to write the names for those, and then we'll go over the lab report stuff. All right, so for the first one, we've got lithium and iodine, so that's definitely a ionic compound or an ionic compound. All right, so we got lithium, we named the metal first, okay, we named the non-metal second, and we ended in ide. Nothing special about that one, it doesn't need Roman numerals or anything like that, just lithium fluoride. Okay, for the second one, we've got calcium, which is a metal, so again, this is an ionic compound, so I just write the name of the metal, and then I write the name of the non-metal. In this case, it's the polyatomic ion, sulfate, whose ending I do not change. Okay. And then for number 5, I have aluminum and bromine. So again, aluminum is a metal, so this is an ionic compound. So I write the name of the metal, the old-fashioned, the way that sounds right in my head way, okay, and the name of the non-metal. Okay, so aluminum bromide.
Is it
Oh, it is, yeah. I don't know why I put fluoride on there. That's weird. You're absolutely right. Maybe I should drink more of my coffee so that I'm more alert and not making silly mistakes like writing fluoride instead of iodide. Okay, for number 8, one of our molecular ones here, okay, which is pretty easy. You probably knew the name of that stuff without having to look it up. What is it? Carbon dioxide. Yeah. Okay, and then we've got 11, right, which is, this one's tricky because it's got that weird polyatomic ion in it. What polyatomic ion does it have in it? Ammonium. NH4 is ammonium. Okay, so this is an ionic compound. Ammonium acts as the metal because it's positively charged, and it is with chlorine. Okay. And then for number 12, we've got sulfur and oxygen. So this is a molecular compound, okay, and will require prefixes, but not on the sulfur because there's only one of it. But what prefix needs to go on the oxygen? Tetra. Okay, how are we feeling about this stuff? Okay? Looks like you guys did pretty good on the quiz, so I think we can, we'll come back to it and we'll play around with it here and there over the next few days, but probably past Monday, we won't spend much more time on naming. So if it is something you want to work on a little bit, okay, you can come and see me for help, okay, or you can work on the worksheets in the workbook because you have the keys in Google Classroom, and then if they're giving you trouble, come and see me. Okay? All right, so you need to take your phones out, okay? And you need to open up Google Classroom, put in there in Google Classroom a lab report template. If you don't have your phone here or you can't get signed in, whatever, that's fine. It's going to be up on the board, okay? But some people like to follow along that way, right? And that is totally fine, right? So like I said, I put it in Google Classroom, which may or may not open. There we go. Okay, so period 1, and it is under the class work tab. It's also there in the stream. Okay, lab reports is where you need to go. In there is a PDF document. Okay, that is going to be what I have on the board here in just a minute. Okay, so lab reports, okay, at the high school level are going to be a bit more in depth, okay, and a bit more detailed than you're probably used to from junior high. Okay, they are obviously still going to have all the same parts, right? Our lab reports start out with a problem. Okay, the expectation for you is that you will write the problem. in your own words. We'll talk about it when we start a lab. We'll say this is what we're investigating, okay? Kind of talk about it a bit, and when I do that, that's talking about the problem. All you have to do is put that in your own words, okay? So let's say I was talking to you about doing a lab where I would see what effect sunlight has on plant growth. I know that's like terribly simplistic, okay, but let's say that's what we were doing, all right? So the problem would be, how does sunlight affect plant growth? Or does sunlight affect plant growth, okay? Something along those lines. It's not a long, okay, sentence or anything like that. It's a pretty simple question, all right? Everybody okay with that? Okay, so that'll be something you're expected to do on each and every lab report. Now, just so you know, okay, when you get a lab in this class, you will get a Google Doc that you are going to write the lab report in. And it is a template, so it's already set up, okay? It'll have problem and it'll just be blank, okay? And then it'll have design, and they'll say manipulated, responding variable, controlled variables, and they'll be blank, but they'll be there. You just have to fill it in, okay? That is the document I want submitted. So each time we do a lab, that will be there for you, a Google Doc for you to fill in, okay? It is the only file I will accept, okay? Don't do it on another program, okay? Or submit some PDF that you made to me, okay? This and this only. Here's why. Unfortunately, I don't want to say cheating, collaborative effort, okay, has been a bit of a problem, okay, over the past few years, okay? And so I make this Google Doc for you to A, save you work, B, make sure you don't forget any parts because they're all there and something needs to be written for all of them, and C, so I can see everything you do when you write this lab report. Now, if that sounds creepy, it probably is a little bit, okay? But because it's my file, I can actually go into it and see all the things that you have done when you produced it, okay? Basically, it tells me, did you copy it from somebody else and paste it in all at once, okay? Because I've had people who, oh, Mr. Cooder, I don't have my lab report done like 5 minutes before class, yet somehow, suddenly, amazingly, they're got it all done and handed in on time 5 minutes later, okay? And all I do is go into the revision history and see, oh, look at that, it all appeared all at once because you copied and pasted it from somewhere, okay? So that's the reason that is the only file that I will accept, okay? It allows me to ensure that you are doing your work, okay? Everybody's lab report, when it gets handed in, should be unique. Are there going to be some things that are similar? Yes, that's okay. It's all right to work with your lab partners or your friends or whatever, as long as you don't just go, I'm going to do the hypothesis, you do the analysis, and we'll copy it and paste it in there, okay? That's not how it works. Then you're missing out on some of the learning that's supposed to go on, okay? So that Google Doc is the file that gets submitted. Okay, back to this. The problem, okay, is the question that we're investigating, the thing we're performing the lab to find out about, okay? What is the effect of light on plant growth, okay? The next thing you'll have is the design. In the design is where you tell me about the variables in the lab, okay? The expectation here is that you will do basically 2 things for each variable. You will identify the manipulated variable. You'll tell me the manipulated variable is this, okay? And then you will explain to me how it's being manipulated and why it's being manipulated. So there's an explanation, okay? There's identification of the variables, and then there's explanation of the variables that tells me a how and a why. So if you do a good job of this, you're looking 2-3 sentences for each variable. Okay, so for this one, because it's a very simple experiment, the explanation is not very long, okay, but if we're investigating what the effect of light is on plant growth, okay, then as the experimenter, I'm probably manipulating the light, okay? I want to see what the effect of light is on plant growth. Plant growth is going to respond to light, okay? So I'm going to manipulate light, right? So in this manipulated variable here, it's going to say the part of the experiment, the manipulated variable is the part of the experiment changed on purpose by the experimenter. What is being tested? Okay. For example, the amount of light given to plants is the manipulated variable because all subjects will receive different amounts of light and light powers photosynthesis. Was there a how and a why? Okay, I said, here's the manipulated variable, it's the amount of light. I'm going to make sure every single plant gets different amounts of light. There's the how I'm manipulating it. Okay, and the why is because light powers photosynthesis. Okay, and I didn't need a whole lot of writing to get that point across. Okay, everybody follow me there? All right. We already said that in an experiment where I'm manipulating the amount of light, plant growth is going to respond to that. Okay, hopefully the more light the plant gets, the more it will grow. Okay, so my responding variable is whatever changes as a result of the manipulations made by the experimenter. Okay, in this case, the growth of the plants, because plants getting more light have more energy for growth. Okay, there was a how and a why. Okay, how is it responding? They're going to grow more if they get more light because light's what they need. Okay, everybody following so far? Okay, this is probably a bit more detailed than you had to do in junior high, yeah? Probably in junior high you just had to go, it was light, and then it was plant growth, and then I just made sure the temperature stayed the same and gave it all the same amount of water. And that was it, okay? All right, in every lab report, okay, you only ever have, in any experiment, one manipulated variable. Okay, why? Why can there never be more than one manipulated variable?
Because you won't be able to tell which different variables affecting what.
Exactly. Okay. If I test more than one thing, I won't know which one of them caused the plants to grow better. I have to isolate one thing that I'm going to change. Okay. That's where controlled variables come in. Are there things other than light that could affect the growth of a plant? Okay. So if I don't want them to affect my experiment, they need to be controlled. That is, kept the same for all the subjects. Okay, so in this experiment, that would be the same kind of plant. Okay, I don't want to compare literally apples to oranges, okay, or carrots to pine trees. Okay, I have to have the same kind of plant. Okay, I would probably need to make sure they all got the same amount of water. Water affects plant growth. Okay, I should probably make sure they're all in the same temperature. Okay, I don't want one that's cold and one that's warm. Okay, that's going to affect their growth. Maybe the type and amount of soil that they're growing in could be a factor that could affect them. Okay, there's lots of things that could affect plant growth, agreed? Okay, in every experiment, you are required to identify 3 controlled variables. Okay, 3 controlled variables for every experiment. There's going to be more than that, and that's okay. You just have to identify 3. Okay, whatever 3 you can find, you can think of. In those controlled variables, you do the same thing you do for all the others. You identify it, and you explain to me how you're going to control it and why you're going to control it. All right, we've just identified a bunch of things we need to control. Okay, so controlled variables are the other factors that could affect the outcome of the experiment if left unchecked. So, examples, same species of plant. Okay, because different plants grow at different rates. Same amount of water, because water is an important factor in plant growth, so each plant needs the same amount, so it can't affect the growth rate of the plants. Okay, have I said a how and a why for both of those already? Okay, same soil, because plants get nutrients from soil, and different soils hold water differently. Keeping them the same ensures it cannot affect the experiment. There was a how and a why. Same temperature, as plants typically slow their growth in extreme heat or cold. Choosing an effective temperature for all plants ensures it does not affect the experiment. How and why. Okay, have I been pretty thorough there? I think I identified 4 there. You only have to identify 3. Okay, everybody all right with that? Most of the time when we do a lab, there will be obviously only one manipulated variable, okay? There will be usually just one responding variable, but sometimes there's more than one. Okay? Is it okay to have more than one responding variable? It is. It's okay to watch and measure a couple of things, okay, during the lab. Right? Most of the time for us, it'll just be one thing. And then, as I said, you have to identify 3 controlled variables and explain each one. Okay, you do that, you're going to get 5 out of 5 on the design. Okay, here's the ways people go wrong in the design. They just identify variables. They don't explain them. Okay? At best, you're going to get 2 out of 5 for them. At best. Okay? If you, some people do this, they combine the explanation for all the controlled variables. They'll say, this is a controlled variable, this is a controlled variable, this is a controlled variable. They were kept the same so they don't affect the experiment. No, not good enough. Okay? You need to explain each one because each one affects the experiment differently. Yes, you're keeping them all the same, but you're probably keeping them the same in different ways. Okay? Here we said the same amount and type of soil. Okay? We also said the same amount of water. Okay? We have to, each one is explained separately. Okay? If you combine them, you're looking at probably 3 out of 5, maybe 4. Okay? Would depend on how you do on the manipulated and responding variables. Okay? So they all have to be explained, identified. Okay? Questions on that? All right. The next thing that comes after your design is your hypothesis. Okay? Now, probably up until now, you've been told the hypothesis is a guess, which is wrong. Okay? A hypothesis is not a guess. Okay? It is a statement. It says the following things. If this is true, and an experiment like this is run, then this is going to happen, or these will be the results, or the experimenter will be able to do this. Okay? It is a deductive prediction. Okay? It says this is what's going to happen. You have to make it that way. You have to set it up so that you can obviously be right or wrong. Okay? A hypothesis has to be built so that it can be either proven or disproven unequivocally. You cannot make a hypothesis that sits on the fence. Okay? I oftentimes have people, they write a hypothesis and it's so wishy-washy. Okay? It sounds like something you'd hear if you were like watching the play-by-play from like the House of Commons or something. Okay? Where it's like, well, I don't want to make anybody mad, so I'm going to say this and this and this, and then everybody will be happy. Okay? Or I won't offend anyone. A hypothesis has to be set up so you can be right or wrong. Okay? Will you lose marks if it ends up that your hypothesis is wrong? Absolutely not. Most of the time in real science, hypotheses are disproven. Okay? You usually have to go through many hypotheses being disproven before you get one that is proven. Okay? And then you prove it, you got to have other people repeat it. Okay? That's how the scientific method works. Right? So no, absolutely you would not lose marks if it ends up that your hypothesis is wrong. If it's in the right format, okay, and it says the right things, it is okay if it ends up being wrong at the end. Okay? Most of the time for our labs, they're pretty cookbook, so most of the time we're going to get the hypothesis right, okay? Just because our labs are fairly simple compared to what, let's say, real researchers would do. All right, so a hypothesis always contains 3 words. Well, it contains more than 3 words, but it always contains these 3 words. If, and, then. Okay? The if part of a hypothesis is what you as the experimenter believe is true, or it is what you think is causing the thing you're observing. Okay? Or it is how light is going to affect the plants. Okay? Something along, or what does light do for plants? Okay? That's what the if part does. The and part briefly describes the experiment. Okay? Your procedure outlines in detail the experiment. Your hypothesis in the and part just briefly talks about it. The then part of your hypothesis is the prediction part. Okay? It is, this is going to happen. Okay? These are the results that are going to be achieved. Okay? Everyone follow me there? All right. So a hypothesis ends up looking, okay, something like this. Okay? It's an if and then statement that communicates what the relationship between the manipulated and responding variables is thought to be. That's the if part. Okay? Briefly describes the experiment. That's the and part. And outlines the expected results. That's the then part. Okay? So sometimes it's like the then part is sometimes the answer to the problem. All right, so a hypothesis for this one on light and plant growth, okay, would sound something like this. If plants require light for growth, okay, that's what I think is true. Plants require light for growth, okay, and 5 identical plants are given different amounts of light, that's a brief description of the experiment, okay, then the plants that receive the most light will show the most growth. Okay, I said, here's what I think the effect of light is, here's how I'm going to test it, this is what's going to happen. Right? Have I set myself up to be right or wrong? Like unequivocally. I've said more light means more growth. That's what I said. Am I going to be right? I'm actually going to be wrong. Are most plants going to want to be in the light 24/7? They actually need some time, okay, where they're not in direct light. Okay? It's part of photosynthesis we call the dark reactions. It's actually where sugar is produced. Remember we talked about how the electrons have to fall back down, okay, in order to provide that energy, okay, for photosynthesis to occur. It's actually important that plants have some time in the dark. Okay? All right. So, as we said, if, what's the cause of the problem or the relationship being investigated, and briefly describe the experiment, then what to expect if you are correct. And here's something else. We generally try to avoid the use of I, us, or we in a hypothesis. That's a hard habit to break. I see it in hypotheses all the time. Here's why we don't want to have references to a specific person. It means that if someone else is going to repeat your experiment to test and see if you were right, you have to be there, which isn't good. They're trying to, like in an unbiased way, replicate what you did. It's actually better if you're not there. Okay? But if you put, I am going to do this, or I believe this, then you have to be there every time it's repeated. We don't want to have that in there. Okay? Or in the kind of mean way I often say to people, like, I think this is going to happen in their hypothesis. I don't care what you think. I care what you can prove. Okay? And a hypothesis can be proven or disproven. I believe, that's your belief. That's nice, you're entitled to it. Okay? But it's not very scientific. Everybody with me on that? Okay. Okay, then we have a procedure. Okay, your procedure is obviously your detailed, step-by-step, explicit instructions. By the way, explicit in this context means that they are extremely clear and leave no doubt as to what you are talking about. It does not mean you are allowed to swear in your hypothesis. Okay? All right, so your explicit directions for preparing and performing the experiment. If you have to write a procedure, and you do actually for the first lap, you need to write it so that anybody could follow it and not screw it up. Okay? So you got to write it and think in your mind, okay, how could somebody mess this up? What do I need to account for? This is not very nice, but it's true. Could the dumbest person I know do this and do it right? Okay? Which means you have to tell them everything. When you do this step, this is what you do. This is what you watch for. This is what you record. Okay? You have to tell them all of that stuff. Okay? If you don't, they'll mess something up, or they won't measure something, or they won't make good observations. You have to tell them what to do, what to record, what to watch for, how to get the materials, how to clean it up after. Okay? All of that stuff goes in the procedure. Okay? All right. Then you have your observations. Okay? Those can come in a variety of forms depending on the experiment. Okay? Sometimes it's direct measurements. If we've got an experiment where a cart is running along the floor, okay, our results would be how far did it go every second. We'd be measuring it, we'd be marking it off on the floor. Okay? Our results, our observations would be numbers. They would be quantitative observations. Quantitative means has quantity or has a measured value. Okay? The other kind of observations are qualitative. They're things that you observe but don't measure. Okay? So if you mix two things together and it goes from being two clear solutions to being bright pink solid, you would write, okay, well, in the beginning, the two reactants were clear solutions, and after the temperature, well, temperature would actually be a measured value, okay, the mixture was a pink, like, kind of goo. Okay, well, that's a qualitative observation. You can't measure the pinkness of goo. Okay, you just observe that pink goo was formed in the experiment. That's a qualitative observation. Okay, it's okay in an experiment to have both. Okay, in some experiments, like regarding like human behavior and things like that, everything is qualitative. You can't measure somebody's, amount of sadness, okay, or someone's amount of happiness. You can't, that's not something you can measure. You can just say, today patient 522, appeared to be in a better mood and their emotions were well controlled, whereas patient 627 appeared overly aggressive and, tried to stab me with a pen. Okay, like those are qualitative observations. They're equally valid, they're equally important, okay, but they're not something you can measure. Okay, everyone follow the difference? So, observations can be charts, graphs, or any other data, including anecdotal notes, okay, acquired during the experiment. So, anecdotal just means you kind of told the story. Okay, this is what I observed, or in my conversation with patient 202, they expressed this and this and this. Okay. All right, in your analysis, okay, most of the time in Science 10, your analysis is going to be, here's some questions to answer. Okay, or here's some calculations that I want you to perform with the measured data that you had. Okay, stuff like that. You'll be told how to analyze the data you got. That's not a true reflection of how real science works. Okay, when you get into like university and you start doing real research, analysis is quite a bit different. Okay, you have data and you have to read it and you have to figure out what it says. Okay, nobody's going to go, hey, you were doing this incredible research on human behavior. Here's how to analyze the data. No one's going to tell you that, okay? In high school science, we do, right? But in real research, you have to figure it out, okay? What does the behavior of my 725 patients say about the drug I put them on, okay? Or something along those lines, okay? Okay, so if it's calculations, you show me all the calculations, you compare the data, you tell me what the data says, okay? Otherwise, it could be explanatory answers like, in every cell that was observed under the microscope, it was found that there was a nucleus in every cell. If there's a nucleus in every cell, that must mean the nucleus is very important to the functioning of all cells. That would be analysis. Okay, that was your data. That's how you would analyze it. Everyone follow? Okay. Then you'll have a conclusion. Okay, in your conclusion, you're going to restate your hypothesis. You can literally just copy and paste it. Okay, so take it from the top of your experiment, copy it, paste it back down. Okay, this was the hypothesis, and now I'm going to tell you whether I can accept it or reject it and why. The why is the most important part. Okay, in the why, you tell me what did your experiment reveal, what did your analysis say about that, and how does that relate to your hypothesis? Does it support your hypothesis or not? Okay, so let's say that in this experiment on plant growth, my 5 plants went like this. Plant 1 was in total darkness 24 hours a day. That's my control plant. Okay, I need to know what happens if a plant gets no light at all. Then I had a plant that got 6 hours of light per day. I had a plant that got 12 hours of light per day, a plant that got 18 hours of light per day, and a plant that was in complete sunlight all day, 24 hours a day. Okay? Those are my 5 experimental plants. My data says this. Plants 1 and 5 are dead. Okay? Plant number 2 showed minimal growth. Maybe I actually measured and I said it only grew 10 centimeters over the 3 months it was observed, and it had 25 leaves on it and 1 flower. Okay? And then the plant that got 12 hours of sunlight grew 35 centimeters. It had 55 leaves and 6 flowers. Okay, and the plant that got 18 hours of sunlight, okay, grew 75 centimeters, had 100 leaves and 18 flowers. I don't know, I'm just making this stuff up. Okay, and then the plant that got 24 hours of light is dead. Okay, if I analyze that data, I do see a pattern. There is an increase in growth with light
to a point. Okay, it does seem like 18 hours of sun and 6 hours of darkness is very good for plant growth, but 24 hours of sun is not. My hypothesis said more light means more growth. My data does not support that. Okay, that's what my experiment says. Therefore, the hypothesis is rejected. Okay, everybody all right with that? Okay, you're not going to lose marks if you have to reject your hypothesis. Okay, you'll lose marks if you accept one that's wrong. Okay, that you would lose marks for. Okay, if you say, here's all my data, this is what it says, it doesn't support your hypothesis, but you go, I like my hypothesis, so I accept it anyway, then you're wrong. Okay, you can get attached to a hypothesis, so you have to be careful about that. Okay, everybody follow on the conclusion? The conclusion's going to be a bit of writing. Okay, there has to be some explanation there. I talked for quite a while about the experiment, the data, and whether or not it supported my experiment.
Okay. All right, so what this would look like here, what I wrote was, as it was found that plants receiving small amounts of light and very large amounts of light showed little growth, it can be concluded that plants require certain amounts of light and darkness to be healthy. Therefore, the hypothesis is rejected. Okay, the last thing you got to do in a lab report is identify 2 sources of error and suggest improvements. Okay? So you're not going to hurt my feelings, okay, when you identify these sources of error. Okay? Our experiments are designed to have error in them for you to identify. So let's say that in this experiment, okay, I was using like a Thomas Edison style light bulb in all of the boxes. I had every plant in its own box, okay? And I had an incandescent light bulb in there. Do incandescent light bulbs get hot? Okay, is my temperature going to be perfectly controlled then? No. Okay, so I might say it was found that the plants receiving more light also had slightly higher temperatures in their growth boxes. Okay, this could have affected their growth because temperature can affect the rate at which photosynthesis occurs. Right? So that's a source of error. How do I fix that? The next time maybe I do this with LED lights, okay, because they don't get hot. Right? Or I have some sort of temperature control in all of the boxes that's going to pump in cool air or warm air to make sure that they stay the same temperature. Okay? My solution to the source of error doesn't have to be, let's say, super realistic. Like, if it would require millions of dollars to, correct this source of error, that's okay. We're not really going to do it. Okay? You identified it. You said this is how I would fix it. Okay, great. If there was unlimited funds, we'd do it that way. Okay? Everyone follow on that? Okay? So you need 2 sources of error and ways to fix them. So a second source of error in that maybe could be that the plants are not genetically identical. They were all grown from seeds. There are obviously individual variations in genetics that could have said that this particular plant was a more vigorous grower than this plant was. Okay? In the future, all the plants should be grown from cuttings of the same plant so that they would be genetically identical. Okay? There's a source of error and a way to fix it. Okay? Everyone kind of follow what we're looking for there? Okay. So the first time you do a lab, it's going to take you a long time. Okay? And we're giving you basically a full class period next week to do the pre-lab stuff, to do the problem, the design, the hypothesis, the procedure, okay? And then the analysis will actually be pretty quick. Okay? You won't get class time for that. Okay? So your first lab's going to take you quite a while because it's your first one. And you're writing a procedure for it. After that, as you get better with them and you're not writing the procedures anymore, a lab report shouldn't be tons of work for you at home. Okay? If you are spending more than, let's say, 40 minutes at home after the class time that you've received, you might be doing something wrong. Okay? I know there are some people out there, they just really are proud of their work and they really want to do a good job and they don't care that they're spending an hour and a bit, okay, or 2 hours writing a lab report. I mean, I've had some kids who hand in lab reports and I'm like, oh my gosh, I could not have written a lab report this good. Okay, and that's fine. If you want to do that, you go right ahead. Okay, but realistically, a good lab report, okay, can be done within the class time plus, let's say,
half an hour to 40 minutes. More than that, and you might have just kind of lost track of something and you've kind of gone sideways. If that happens more than once, you should come in and see me and say, you know what, Mr. Cooder, it's taken me a really long time to do my lab reports. Can you help me kind of streamline what I'm doing so I can work smarter, not harder? Okay, and that's no problem. Okay, but lab reports are not meant to be something that is like 6 or 7 hours worth of work. Okay, they're not at all like that. All right. When you do a lab report, you can bring it in and have me look it over before it's due. Okay, so when we do our lab next week, okay, obviously it won't be due before the break, it'll be due after the break. Okay, but I'll make sure there's a couple of days there after the break where if you want to, you can come in and see me before school or at lunch or after school and say, Mr. Cooder, can you quickly look over my lab? Okay, it won't take me more than about 5 minutes to look over your lab and give you some feedback. Okay, when I give you feedback, unless like you're totally off base on your lab report, I might send you home with 10 minutes worth of edits. Okay, probably not even that much. Okay, sometimes people bring in a lab report, Mr. Cooder, can you check it over? Yep, it's 30 out of 30, submit it. Okay, that happens. Okay, sometimes I'm just like, that's perfect, hand it in exactly as it is. Okay, and other times I'm like, you know what, you kind of missed the boat on question number 2. Can you talk maybe about this stuff in your answer instead? Okay, or can you add a little more detail to your hypothesis? I'm looking for this a little bit. Okay, stuff like that. It's not going to be huge amounts. Okay, lab reports, okay, we're gonna have a few of them, guys. We do 2 or 3 per unit. Okay, so they do contribute to your mark. So, tomorrow, plan for tomorrow, guys, we'll do a little bit of naming practice, and then we're probably gonna start looking at properties of compounds, okay, which is what our lab next week is gonna be about. Okay, good work today, guys.